I want to thank the organizers for inviting me, and thank you for the nice introduction. I hope I can live up to it. I'm thinking that I'll say from the outset that a lot of the work that I'm going to be talking about, actually all of my thinking, is very much inspired by work that Bill Newsom has done. So it's really nice to be here, and maybe he's going to hate all of it, and I'm sure that he'll let me know if he does. I'm going to start. I'm trying to see if I don't have a mouse pointer, so that means that I'm going to end up using the laser pointer, and I'm going to end up using it on one screen. Sorry for that. Mouse doesn't show up. So I'm going to start with an old picture of the macaque-monkey visual system. This is different. Each of these boxes, there's a different region. Is it OK if I use this one? OK, but this one has the slide advancer. All right. We're going to test my sensory motor coordination. So each of these boxes is a cortical region, and it's an old picture, 25 years old, but it's still very much used in talks. And when they developed it, the idea was to lay out the organization of the visual system and to help us understand it. But I think most people use this slide in the way that I'm going to use it here, which is really to point out how complicated the brain is, and to say that the brain is really dizzyingly and bewilderingly complex. You look at the wiring diagram here, and it's this big spaghetti mess. I'm going to say that in Barbara's talk, and Barbara will correct me if I'm wrong, there were symmetries that could help you think about the modeling and the circuitry. But here it's not obvious that there are those symmetries. Each of these regions could have very different properties, and of course each region has many different types of neurons that each of those could have very different properties. So as everybody in this room knows, the problem of understanding how this works is really extremely difficult. And if you're interested in cognition and in the whole pathway from sensation to cognition to motor action, the problem is even harder. If you're focused on perception, you might focus on sensory areas. If you're focused on motor action, you might focus on motor cortex or other motor regions in the brain. But if you're interested in that whole pathway in a behavior, for example, if you're interested in perceptual decision-making, you're going from the perception to the action. You need to think about the dynamics of this extremely difficult and complicated circuit. So that raises the question, why do neuroscientists look so cheerful? And I have to point out, look, they're all smiling. Okay? And so you've heard some of these talks, so you know why these particular individuals are smiling, and they have very good reasons. But I'm going to claim that neuroscientists as a whole are smiling. So what is it that is making us so happy? And of course, you guys know some of the answers to this, and it's the enormous methodological revolutions in the last few years. I'm going to mention some of them, transgenic lines that allow you to identify specific genetically defined neurons, optogenetic. Since I'm at Stanford, I thought I'd put a picture of Carl Dijkstra and show that he's smiling too. And so this allows you to perturb specific neurons with enormous time specificity. The advent of large-scale recording. This is a picture from David Tank's lab showing calcium imaging of 350 simultaneously recorded neurons, and that number keeps growing and growing. And it's not only the calcium imaging, but electrophysiological methods. For example, these are some of the new probes, silicon probes coming out of Janelia, collaborating with other people, where, again, several hundred neurons are being recorded simultaneously. So our ability both to obtain data very specifically marked as to what neurons we're recording, and our ability to perturb the brain in very specific ways has changed radically in the last 10, 15 years. But I'm going to claim that while these things are a very good reason for a lot of neuroscientists to be smiling, they're not enough. The problem is still really hard. And so I'll tell you about a couple of things that my lab is thinking about to try to address some of the problems that remain. In particular, if you're working if you're thinking about cognition and you're working with animals, you're not working with humans, so you can't just tell the animal what you want it to do. You have to train it. Often, this is a very time-consuming step, and I'll tell you about some of the things that we've done to try and get around that. And the problem with thinking of the dynamics of this circuit, that remains. We can record and we can perturb all these neurons with exquisite specificity. But how does this extremely complex circuit work? What is the dynamical interplay between activity in all of these neurons and all of these regions? what is it that leads to cognition, perception, thinking, speech, sitting and listening in a talk? Still a very difficult problem. I'm not going to tell you any answers. I'm going to tell you about some of the approaches that we're trying to pursue. So on the behavioral training, this little corner, I'll tell you the story in the following way. I like telling it thinking about how cognitive questions were typically addressed about 10 years ago, and some things have changed. And here I'm specifically talking about cognition, people who are interested in studying it at the single neuron level, where we want single neuron resolution of the activity of the neurons that is related to cognition. And I'm saying that because, of course, if you're working at a much broader level of resolution, you can use fMRI, you can look across the brain, you can do enormous recordings. But at the level of single neuron resolution, it's mostly experiments with animals. And it was mostly experiments with monkeys and what people would do. And this is very beautiful work, and it's the kind of work that I was alluding to that has inspired me so much. People would develop some particular well-controlled behavioral tasks that would try to isolate a particular cognitive process that they were interested in. And then over a period of a few months, train a few monkeys to perform this task. And then carry out electrophysiological recordings to see what the activity of neurons during this particular cognitive process looks like. And then maybe carry out some causal perturbation studies. This typically happened less often because, of course, after you've invested so much time in training, these animals are extremely valuable. And the very nature of a causal perturbation is that you're going to go in and muck with the brain. So that's something that your natural tendency is going to be very conservative and careful about that. And I know that there are many people here carrying out research in this style, which I think is very beautiful research. And if I say things that annoy them, I hope that they'll point it out and challenge me. And as a postdoc, I worked with Ranulfo Romo in Mexico, and he was doing some very beautiful experiments that I would describe as being in this style. And he was recording from monkeys that were performing a working memory task. And he got some beautiful data from prefrontal cortex and many other regions. And when I started my own faculty job as a computational neuroscientist, I was very interested in trying to produce neural network models of how this kind of activity came about. And we produced such a model. And since I'd been involved in making both this figure and this figure, I can make the colors match. And if the colors match, these two things must be related, of course. But we were very happy with the picture. And then, of course, we wanted to test the model in some way. And we figured out some experiments that we thought would test the model. And so I went to talk to Ranulfo, and I said, oh, here are some experiments that we think would be great to do. And he was super nice about it and super interested and very enthusiastic. And he said, that sounds extremely interesting. Let's definitely do it. I have one experiment that I want to do first, so we'll get to your experiments in about 3 or 4 years. And that just seemed like a tremendous bottleneck, right? And why was he saying that? Well, the reason was these numbers, of course. Right? That's the kind of time scale over which these experiments turn around. And that felt really extremely difficult. So together with 2 other labs that had started doing this at Cold Spring Harbor, Tony Zader's lab and Zach Mainen's lab, we set out to try to automate the process of behavioral training. And to do that, working with monkeys was too difficult. They're large, they're expensive. We thought that working with rodents would be easier. And we started, my lab still mostly worked with rats. There are, of course, many labs that are now also doing this kind of thing with mice. And the idea was to train the rats and to treat them with the same kind of quantitative care that we'd learned from these guys, and with the same kind of quantitative and rigorous thinking, or at least that was our goal. And of course, they're small, they're cheap, we could get good behavior out of them, and they're amenable to modern molecular tools. So this is a picture of one of the walls in the behavior room in my lab, showing you 9 out of 30 rigs that we have in the lab total. This is a computer rack, so a person is about that tall. And we have technicians that move the rats around. And in each of the rigs, we train 9 shifts per day, so we train 9 rats per rig per day. So in total, we're training about 270 individual rats every day. Whoa, that was not what I intended. Fine. So a typical student or postdoc in my lab supervises about 30 animals. And of course, if you have 30 animals that are all training, there's no possible way that you can sit with each individual animal and shape it carefully and do that kind of artisanal training that is often done with non-human primates. So that forced us to develop automation of the training process. And so to do that, we spend a lot of time figuring out, for any particular task that we're working with, what is the computer code that will take the animals, take a naive animal, all the way through to the final behavior that we need to do. And developing that code can take a long time. They often do things that you don't expect, and then you need to correct for that. But once we have that code, then it's beautiful, because then we can parallelize up the wazoo. As an example, typical behaviors that we use in the lab, we're interested in these, what we'd like to call higher level cognition tasks. They're tasks that are difficult to train. So they typically take about 4 months of training per animal. But if we take 40 out of these 270 slots and dedicate them to a particular task, then on average, we get 1 trained animal out every 3 days. And that's a completely different world to having 2 or 3 animals a year. And in particular, we like to think that it allows us the freedom to try stuff. And one of the kinds of things that it allows us to try is to try causal perturbations. There's a new reagent, let's just try it with our trained animal. If it doesn't work, it doesn't matter. We can go again. One of the things that we're doing is that after we publish a paper about a particular task, we then post the training code on the web. So in principle, that means that anybody would now be able to reproduce the behavior exactly. It's very time-consuming to figure out the training code for one of these behaviors. But if we post it, and the hardware is reproducible and the code is reproducible, then in principle, the behaviors should be exactly reproducible by anybody, anywhere in the world. Every time I give this talk, I hope that somebody will take us up on it and download the code and run it and train an animal. So far, it hasn't happened, but at least in principle, it should be doable. So some of the reasons why people in my own lab are smiling are this kind of semi-automated behavioral training. People can order rats by email. We assign them into the different training slots through a web interface. They can monitor the training through the web. And then after a few months, they have their trained animals that they can then work with. So I like to call this, you can train in your pajamas, because basically you do it from the comfort of your bed. So that makes them very happy, especially say this particular guy has a baby, lives about an hour away, so he's very happy that he has a good family life and he trains tons of animals. And we're also smiling because we're trying some new computational methods to explore that very complicated circuit function. And this is what I'm most excited to tell you about today, because it's very new for us. This is something that's ongoing. I'm not going to give you any answers at all. I'm just going to point you to the kinds of things that we're trying. So I'm going to tell you a story centered around a particular cognitive process, executive or cognitive control. We have others going on in the lab, but today I'm going to focus on this one. And very loosely speaking, you might define cognitive control as the ability to control how you think, the ability to control how you respond to a stimulus. The example I like to give is that I could tell you, today, red means go and green means stop. And you would be able to go out and drive like that. And it would take you some effort, but you would be able to do it. So somehow, there's something in your brain that can control the link between the red stimulus and whether you press the accelerator pedal or the brake pedal. And that control is under your conscious capacity. It's called top-down control. So how does that work? How does the brain implement that? And the oldest lab task that I know that has this kind of feature is the famous Stroop task that I imagine perhaps all of you have heard about before, where you can take a subject and you can ask them to read the text of a word, red, orange, blue, or you can ask them to name the color with which the word is written. Here it would be green, blue, red. And just like that, just by telling the subject, I want you to be reading or I want you to be color naming, you can take one stimulus and have the subject produce 2 completely different responses. And the switch between those 2 is extremely fast, right? A couple of 100 milliseconds, as soon as you tell the subject what you want them to do. So how does that switch happen so fast? It's too fast for it to be changes in gene expression. It's too fast for it to be changes in structural connections between neurons. So we think that what it is changes in the electrical activity of the neurons. And this is what we'd like to try to understand. In the Stroop task, these 2 different kind of rules have asymmetries between them. Reading, perhaps because we've spent so much of our time reading words, is something that people do very quickly. They do it very easily. They make very few mistakes. They respond very fast. And people often refer to this as the prepotent task. Whereas color naming, that's something that you're much less used to. So people make more mistakes. They respond more slowly. And this is thought to be a slower well, it is a slower, the data is that it's a slower task, and it's harder in the sense of higher percentage of errors. So we wanted to find a task for rodents that would mimic some of these properties so that we could study this in the rodents, and rats in particular. So then we could, our whole program of, oh, we're going to parallelize and we're going to study lots of things. Let's see if we can get that to work. It took us a long time, about 4 years, to find the tricks to write the correct computer code that would train rats efficiently in this task. But now that we've got the code, it takes about 3 months to train each individual animal, and anybody can do it, right? Hopefully the idea is that we put in all this pain so that you don't have to, or at least that's the plan. So let me take you through a trial of the task. Time is going to run horizontally. The rats are in a booth. I'll show you a little video of a rat doing this. So they're in a little behavior booth where they face 3 nose ports. That's little openings that they can stick their nose into. And the very first thing that happens is that in that center, there's 3 of them in a row. In the central nose port, a light comes on. And that's a signal to the trained animal to put their nose in there and keep their nose in there until we turn the light off. We call this the nose fixation period. And when they put their nose in there, we play one of 2 sounds. On any trial, the sound will either be what we call the pro flavor, or it'll be what we call the anti flavor. And the pro sound sounds like wong, 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 wong. And the anti sound sounds like blah, 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 blah. And I make those noises just to emphasize that those 2 sounds are very different sounds. We don't think that the rat has any difficulty in distinguishing between them. We play the sound for 750 milliseconds. Then there's a 500 millisecond delay. And then we turn that center light off, and the rat withdraws from the center port. And at that point, we turn on an LED either on the left of the rat or on the right of the rat. And here comes the interesting part. If the previous sound, or the sound that we played at the beginning of the trial, was pro, then what the rat has to do is turn towards the LED. If the LED was on the left, go left. If the LED was on the right, go right. But if the previous sound that we played was the anti-sound, then the animal has to do the exact opposite. If the LED is on the left, now go right. If the LED is on the right, now go left. And the thing that was difficult to train the animals to do was to be able to switch between those 2 on a trial-by-trial basis. It's not relearning from the pro rule to the anti rule, but on a single trial basis, it can switch from one to the other. It knows both, and it's able to switch rapidly. So let me show you a video of 3 successive trials. We've slowed the video down by a factor of 2. So here's the rat. Sorry, that's the pro sound. Light turned on the left, and the rat turned to the left. Let me show you another pro trial. That tells it go toward the light. Okay, off it goes toward the light. And now this is going to be an anti-trial. Rat almost goes towards the light, but then eventually it turns back and goes the other way. So there's a couple of things that I want you to notice from that video. First, that there are no obvious differences while the rat has its nose in the center. There are no obvious differences between the pro trials and the anti-trials. I emphasize obvious. In the lab, we often refer to the butt-cheek hypothesis, which means that since we're not recording EMGs from every muscle, we don't know if the rat internally tenses its right butt cheek during pro trials and tenses the left butt cheek during anti-trials. We don't know that, right? So that's why I emphasize no obvious differences. The other thing that you saw was that hesitation before the anti-response. The video is a little atypical in that in a typical trial, in a typical anti-trial, the rats don't obviously turn towards the light and then turn away. But what is very typical for these trials is that they're slower in their anti-response. The time they take from the turning on of the LED to their response is slower for anti-trials than for pro trials by about 50 milliseconds. Okay. We compared the behavior of the rats in this task to similar tasks in primates. And it turned out that we found a lot of similarities. So first, the one that I already told you, that pro is faster to execute than anti. We also found, I haven't shown you the data for this, that pro is easier to learn than anti. If we ask them to learn just one thing, just pro, versus learn just anti, pro they learn in 1 session, anti it takes them many sessions to learn. The performance, the percent correct on pro is higher than on anti, especially when the 2 are intermixed with each other. And if we run the trials in little blocks, say a sequence of anti-trials followed by a sequence of pro trials, switching from anti to pro is harder than switching from pro to anti. And that's an asymmetry that we didn't expect. We thought that switching into the hard task would be the hard thing, but it's actually the other way. And it turns out that all of those are very similar to what is seen in primates. Even that delay, the anti is slower by 50 milliseconds, that's also what you find in primates, and a similar number, about 50 milliseconds. And the asymmetry, you also find that asymmetry in the Stroop task. It's harder to switch to the easy task than to switch to the hard task. So we don't know whether the circuits that implement this kind of thing in the rodent are the same as the circuits that implement it in the primate, but at least there's enough behavioral similarities that we thought that there would be a fruitful conversation across the species. And in particular, because of the similarities, we thought, well, let's look at the primate literature, and let's see if there's something there that will inspire us as to how we think the rodents do it. And indeed, there is. And in particular, there's this idea in the primate literature that information about the LED is taken to appears in the superior colliculus. This is particularly plausible for rodents. 75% of retinal ganglion cells in the rodents project to the superior colliculus. Many of those also project to the LGN and then to visual cortex, but there's a strong projection directly from the eyes to the superior colliculus. And the superior colliculus itself, the deeper layers of the superior colliculus, are known to control contralateral motion. So for example, if you activate the left superior colliculus, you'll get a right-orienting motion. And if you activate the right, you'll get a left-orienting motion. So this is a great candidate for the pro circuit, how our pro response is implemented. And it's a great candidate because there's a strong projection. A light on the left would activate the right superior colliculus, producing the leftward motion. It's a very short circuit. It doesn't even go through cortex, so you get very quick responses. And the idea is that information about an anti-trial, about the rule, this is an anti-trial, through pathways that I'm not describing, would go from the sound to prefrontal cortex, and prefrontal cortex would inhibit this prepotent response to then later in some way implement the anti-response. Okay? That's the hypothesis. And so we started thinking, well, let's test that. We think that all of the interesting computation is going to be happening in prefrontal cortex or perhaps other frontal cortices. In this picture, the superior colliculus is really mostly kind of an output motor pathway. It's just the default pro response. And so we thought we'd do some experiments in the superior colliculus and the prefrontal cortex. And the superior colliculus, we almost thought of it as the control. This is where the boring, simple stuff is going to be happening. That's not what we found. The very first thing that we did was use pharmacological inactivation of the superior colliculus on one side. And if you do that, we found results that were very consistent with what people had seen in cats and primates. Namely, if you inhibit the right superior colliculus, then you've inhibited the animal's ability to go left, and you get a big ipsilateral bias in this kind of task where the animal has to choose one or the other. So inhibition of one side of the superior colliculus, big ipsilateral bias. But what we did then was something that had not been done so much in the primate literature, which was simply inactivate the superior colliculus on both sides. And what we found there, to our surprise, is that when we did that, instead of completely disrupting the pro response now became rescued in the sense that, in terms of percent correct, there was no longer any significant deficit on the pro trials. The animals are a little bit slower. Their reaction time is slowed. But they were essentially, in terms of percent correct, they were essentially intact for pro. And in contrast, there was a big increase in errors for the anti-trials. Completely not what we expected. So we've ended up doing a lot more work about the superior colliculus than we were thinking we would do. So what I've shown you so far is something that is published. And what I'll show you from now on is the stuff that we're working on right now, unpublished work. And with the pharmacological inactivation, of course, we get a very slow time scale. We're really comparing inactivation sessions versus non-inactivation sessions. So I think this was one of the very first viruses that we got directly from the Dijsselhoff lab. And I think some of the first experiments still use the batch directly from them. So we used the virus that expresses halorhodopsin under the control of the CAMK2 promoter, and we injected it into the superior colliculus so that now we could use light to inactivate the superior colliculus and get a temporal resolution on inactivation on the order of tens of milliseconds instead of tens of minutes. Okay. The very first thing that we did was just select a subset of full trials to inactivate, a randomly chosen 20%. And what we saw there was that the results that we'd gotten from the pharmacological inactivation and from the optogenetic inactivation were pretty much the same, meaning there was no effect on the pro trials, but a big increase in errors for the anti-trials. But of course, our interest was not in doing the full trial inactivations. That was just a sanity check. But our real interest was the idea that now we could look at subparts of each trial. There's a lot of things going on in each trial, right? At first, the animal has to perceive the rule, then hold it in mind. Then the target comes up, and it has to combine the rule with the target. Very different phases, what's going on in the different phases. So if we inactivate the cue during the cue period, when the pro sound or the anti-sound is playing, we found no effect. And if we inactivate once we've turned on that side light, or what we call the choice period, again, we found no significant effect. But it was when we inactivated during that delay between having played the rule sound and the target light going on, that's when we found the effect. And it was the same type of effect as when we did the full trial. In other words, significantly increased error rates on anti, not significantly increased error rates on pro, and a significant difference between the two. So there's something special that's happening during that delay period. We don't quite know what it is. So we then turn to recordings to try and see, okay, what does activity in the superior colliculus look like during this task? And so I'm showing you a summary of 180 neurons that were recorded in deep layers of the superior colliculus while the animals were performing the task. Each of these rows is a neuron. And what the color represents, if the neuron is coded as very green, that means that at that time point in the trial, this neuron fired a lot more, had a higher firing rate for pro trials than for anti-trials. Whereas if it's color-coded as very red, that means that at that time point, that neuron was firing a lot more during anti-trials than pro trials. And so here we've sorted them according to the timing of the peak difference in firing rate between pro and anti. And what you can see is that about half the neurons are colored green and about half are colored red. There's an intermix of the two. And also the timing of when they have information in their firing rates about whether it's a pro trial or an anti-trial is also pretty mixed. There are some neurons that are early, some neurons that are later, and some neurons that are even later. So it's a pretty mixed representation. You can take those very same neurons and now color code them according to whether their firing rate is bigger if the animal is eventually going to move to the same side of the superior colliculus, to the same side as the side that the electrode was in, or to the contralateral side, to the side that the electrode was in. And we find something similar. There's a lot of information. Once that target light comes on, once the side LED comes on, there's a lot of information in the superior colliculus as to which way the animal is going to move. And there's a big mixture of stuff. You know, about half the neurons fire more during contralateral motion, about half the neurons fire more during ipsilateral motion. And there's no obvious relationship between whether a neuron was a pro-preferring or anti-preferring neuron versus whether it's a contra-preferring neuron or an ipsi-preferring neuron. So what you might think of as a heterogeneous mixed representation. And so we thought that we would be writing a paper with the word heterogeneous in the title. But as it turns out, that's not what we're doing. So let me tell you why. Let's take a closer look at these neurons, and let's just separate them in terms of when they carry information as to what kind of trial it is, whether it's a pro trial or an anti-trial. And we're going to separate them into neurons that carry that information early on during the cue period when the pro or the anti-sound is playing, and neurons that carry that information later. Okay. And if we then try to decode from the population activity what kind of trial it is, we can train our decoder, that is, set the parameters for it based on the correct trials. We take a subset of trials, we take only the correct trials, and that's where we train the decoder. And of course, a decoder that is looking at the cue neurons, the blue neurons, that by definition will have most information as to whether it's a pro or an anti-trial during the cue period, and then that information falls away. That's by the very definition of how we define these blue neurons. But the interesting thing is that if we then look at error trials and we run the decoder on those error trials, we get pretty much the same performance, which is a way of saying that these cue neurons don't behave very differently on trials where the animal performs correctly or on trials where the animal makes an error. So they're not very tightly linked to the behavior. In contrast, if we look at the delay neurons, again, by definition, we're building our decoder based on the correct trials. So of course, there's a lot of information during the delay and much less earlier on. That's by the definition of the delay neurons. But now, if we compare the error trials to the correct trials, we find that there's a big difference between the two. That is, these delay neurons are much more closely correlated to the animal's behavior. If we now ask questions about the information in the superior colliculus about whether the animal is going to go left or right, we find, as I said before, and as you can see from the picture on the top, that of course there's information there, so the decoder is at chance before the side light comes on. Once the side light comes on, the animal is making its decision as to which way it's going to go, and you can see that information reflected in the superior colliculus. But if we divide the decoder to work either with delay neurons or the Q neurons, we find that the delay neurons have that information significantly earlier than the Q neurons. And furthermore, if we look at other regions of the brain, for example, the prefrontal cortex, from a neural activity in the prefrontal cortex, you can also decode which way the animal is going to go, but much later than in the superior colliculus. So these delay neurons, the neurons that have information as to is it a pro-trial or an anti-trial, are tightly linked to the behavior. And that's where you first see information as to which way the animal is going to go, suggesting that the decision might be computed in the superior colliculus itself, certainly not in prefrontal cortex. So that led us to focus on these delay neurons, and focusing on those delay neurons revealed an order in what we previously thought was this random mix of pro versus anti versus contra versus ipsi. And that's, if you look at the delay neurons and choose and select those neurons that, if we ask whether they fire more for ipsilateral motion versus contralateral motion, you choose the ipsilateral preferring neurons, you find that those neurons also have a strong tendency to be neurons that fire more during anti-trials than on pro-trials. And if you now look at neurons that fire more when the animal moves contralateral to the side that has the electrode, those have a strong tendency to be pro-preferring neurons. They also fire more during pro-trials. So rather than a big mix of stuff, it looks like there's really two physiologically defined populations of neurons. The pro-contra neurons fire more during pro-trials and also during contralateral motion, and the anti-ipsi neurons fire more during anti-trials and also during ipsilateral motion. So there are two sides to the superior colliculus. So that means that if we look at these neurons, we're going to be thinking about two sides of the brain. I should point out that because we know that the deep layers of the superior colliculus control contralateral motion, we think that these neurons, the pro-contra neurons, are the output neurons of this circuit. And since we have two sides of the brain, in this most simplified cartoon of what's going on in the superior colliculus, we have at least 4 nodes, each node representing a group of neurons. The anti-ipsi on one side, the other side, and the pro-contra on the two sides. And at that point, we started doing some modeling, perhaps thinking along the same lines as Barbara, as thinking we want to quantify what's going on. We want to come up with a model of how we could reproduce the neural activity and also the optogenetic results. And so we started looking at all the parameters that would go into it. If you have these nodes and you're thinking of neural activity in the nodes influencing each other, you'd want some vertical connection weight between these two, and let's say for symmetry that the two sides of the brain are the same. And let's say, in fact, that the upwards going parameter is the same as the downwards going here. Then there's going to be some connection between the two sides of the superior colliculus, and there might be a connection between the same flavored neurons, or there might be a connection between differently flavored neurons. There might also be, of course, self-connections within each group, so that's another parameter. There's going to be general excitation that is driving the network. There's going to be some strength of the signal from prefrontal cortex that feeds the information as to is this a pro-trial or an anti-trial at the beginning. There's going to be some strength of the input of the target light. There's going to be some intrinsic properties of the neurons, et cetera, et cetera. And by the time you finish listing kind of a minimal set of parameters, we ended up with 15 different parameters. So we scratched our heads and we worked really hard and we tweaked the parameters, and finally we got a set of parameters that kind of reproduced the behavior, and they got the difference in the reaction time, and they more or less reproduced the physiological results, and they also reproduced the optogenetic results. And this felt extremely unsatisfying. And the reason it felt unsatisfying is that we had an example model, but we didn't know what other values of the parameters out there might also be a plausible model. And what we want to do with these models is to guide the experiments, right? Is to use them to think, what's the next experiment going to be? And doing that, we've tried to make the cost of experiments cheaper than before by the automated behavior and so on, but they're still not cheap, right? You're talking about somebody's life for 3 years or so. So were we really convinced that this would lead us to the best experiments to do? No. So how do we explore that space? And I want to point out that here I'm talking about the superior colliculus. That's a corner of this much bigger, much more complicated circuit. In fact, it's such a small corner of it that it's not even in this picture. I'll briefly point out that 10 years ago, we could get away writing papers about 2-dimensional networks, but now the networks are much bigger dimensionals. How do we explore this parameter? One thing that we could do, and Eve Marder has done some work on this, is just explore the whole parameter space, brute force. Okay? So say that each parameter, you sampled it at 3 values. That would be 3 to the 15 different example networks that you want to look at. And with current computers, we rented 2,000 CPUs from Google. This would take us about 5 days. But we don't know how finely we need to sample. So if it turns out we need to sample at 5 values per parameter, then it would take us about 30 years. So a bit of a problem. So what is it that we're trying now? Well, what we're trying now is trying to use gradient descent to try and simplify the space that we're looking at. So let me explain what I mean by that. For any particular set of parameters, we have some measure of how well our model matches the data. And what we like are models that match the data really well. So brown in this kind of schematic. And if we take a bunch of different starting points for the parameter values and follow them downhill in the cost of how good or how bad the model is, well, if we do that, we're going to end up at the bottom of these valleys. And all those different parameter sets, all those different example networks, are really represented by a single network that has the parameters that lie at the bottom of that valley. So that single network represents that entire basin. Okay? And so that means that we can sample much, much more efficiently. For example, if instead of taking a grid of points and exploring each one of those, we only sampled this one, and that one, only 3, we would end up at the very same points than if we had sampled that whole big grid. So the entire basin gets collapsed into a single point. And that makes the exploring of different networks much easier to do. This idea of essentially training the networks to optimize them and looking at optimized network is not really something that's new. And to do it, all we need to do is use calculus to find that gradient. So also that is not new. But what is new is just the tools that are available. So for example, there are some tools that make taking the gradient that make it practically very easy, and also finding the second-order gradient, making it very, very easy. And in practice, that means that our ability to rapidly cook up a network and try it out and do this on it has become much, much easier to do. So now this is something that we can really do. And there are many tools that do this algorithmic differentiation now. We happen to like a particular tool in the language Julia, but there are many other versions that people use. So what we're doing is we take this network, we start from many random starting points, and then we find those minima, and we look at the set of minima that match our data well. I told you that I wasn't giving you answers yet. We don't have any answers, so we have some preliminary suggestions that this will be interesting. It turns out that we find really 2 clusters of solutions. And so the things that we're excited about are that we can now ask questions like this. Out of the space of all possible models, what are the experimental predictions that are robust across all of those? What are the experiments that would distinguish between those different solutions? And those are questions that in practice we could think of asking, but we actually couldn't ask them 5 years ago. And now we can do that. And also that space has collapsed so much that we think that we might be able to scale up to bigger networks, perhaps not something yet as complex as that, but in the kinds of networks that we typically think of in decision making, we're talking about a subset of regions in the brain, not all of them. So maybe when we're talking about 10 to 20 different regions, we might be able to use these methods for that. So to conclude, I've told you about tons of reasons for neuroscientists to smile. Some of the bigger ones are revolutions that my lab hasn't led, but that are out there and that are experimental. And I also told you about reasons to smile kind of conceptually about how we think about the dynamics of networks and about those very complex spaces. one of the things that I pointed out was this whole idea of using rodents to explore cognition in careful ways. I want to emphasize that we think of it as complementary to the research that is going on with primates, not in any way as a replacement. There are things that are easier to do in rodents. There are things that are much easier and much better done in primates, and we're really interested in communicating across the two. I told you that we were surprised. Bill tells me that he wasn't so surprised, but we were surprised that the superior colliculus is part of executive control in this task. It plays a much more cognitive role than we had previously appreciated. And lastly, I told you about some of the latest computer science tools, and for us, algorithmic differentiation. It's not a new idea. It's been around since the 60s, actually, but the practical implementations of it are relatively new, and they're making our life very different and giving us a lot of fun and a lot of reasons to smile, because they're really empowering us to do a much broader exploration of the possible dynamics of circuits that underlie cognition. And I think that's my last bullet point. We're hoping that these methods will scale up to larger circuit. As I told you, I didn't give you any answers. These are just the things that we're thinking of and the directions that we're exploring. And over here are the people that have actually been doing most of the work. Thank you.
Hi, Carlos. That was very nice. A couple of points. One, have you tried inactivating PFC to see what effect you have? I mean, I don't think of superior colliculus as a working memory, sort of persistent activity kind of place, but I guess I could be wrong.
You find lots of persistence activity in the superior colliculus. But the way I think of it is that at the heart of your question is the question, is it that the superior colliculus is doing this alone? And the answer is no, we don't think so. And I'm sorry that I didn't emphasize that. We think that the superior colliculus is part of a broader circuit that is doing this. And so we think that a lot of what we see in the superior colliculus, we might see elsewhere. In particular, we have inactivated the prefrontal cortex. And we also see an impairment in anti-trials, not in pro-trials, when we inactivate the prefrontal cortex. So the way we're thinking about it right now is that the information as to is this a pro-trial or is this an anti-trial doesn't arise in the superior colliculus alone, but we think that as in that primate hypothesis that I sketched out, it comes in from prefrontal cortex, in which case you would see those effects of inactivation that we saw. So that's how we're thinking about it.
Okay, and then on the second, for the radial descent methods, I'm always worrisome about those that you might be missing local steep wells that you might not sample and end up in, especially in this complex 15-dimensional space.
Absolutely, and that is also something that I should have emphasized and didn't. These methods are just helping us get a sense of the space out there, but they do not guarantee anything. If there were a particularly deep, great solution that had a very, very small basin, we would miss it. You know, we might miss it with the grid. We would also miss it this way. So it's just giving us a better sense, but it doesn't guarantee that we found the best solution, not in any sense.
Thank you.
That was beautiful, Carlos. On your question about circuits, I think you mentioned very briefly in passing, but I wonder if you can say more about these two different cell types in the colliculus. Superficial layers do something very different from the deeper layers in the colliculus. I don't know that deeply, but maybe you can comment on whether these two cell types map out to different layers in the colliculus.
No, we don't think they map out to different layers. So the superficial layers are where the visual input comes in. That's where the axons from the retina come in and make their connections. All our experiments, both the inactivations and the recordings, are in the deeper layers. So we think that both of these types of units are in the deeper layers. And we do not have any way to identify them right now other than physiologically from the behavior. So for example, we do not have a genetic marker for pro-contra neurons that would currently allow us to specifically perturb them.
When is this going to end? Thank you, fascinating. I'm interested in the relationship between the actual and the virtual. When will such modeling emerge in conceptually Google Brain connectome at the atomic and cellular levels, in addition to the street view levels in your understanding?
I have no idea. I'm going to guess 20 years just to give a guess, but I don't really know. 20 plus minus 20? I didn't give error bars.
So Carlos, very beautiful as usual. The inactivation of one side of the colliculus, and you suppress orientation in pro, but then you rescue it by suppressing the other side. That's Sprague effect, right?
In a way, yeah.
Yeah. And I've always kind of rationalized that by saying, okay, the colliculi or visual cortices are in competition with each other. You inactivate one, you give a huge advantage to the other, but you inactivate them both, they're on equal ground. So I can kind of rationalize that. But I don't understand why why that same logic doesn't apply for the anti. You said something about a model being, after you tweaked all the parameters, being able to account for the difference in behavior on anti versus pro, and I'd like to have an intuitive understanding of that if one is available, but I understand if there's not.
So let me try and describe it. And first I'll say that our thinking is exactly along the same lines. The fact that the pro was rescued, we thought, oh, it wasn't what we were expecting, but it didn't seem paradoxical. It was the anti-effect that we were most surprised by. So in the model that we managed to hand tweak, the way it works is that there's pro versus anti-selectivity during the rule and during the delay. And then when the target light comes on, we've got our 4 units, and the thing that we hand tweaked in was inhibition between all 4 of those. So it's a 4-way mutual inhibition network. That network, the natural dynamics of that network end up being one where there's 2 attractors, so 2 states that it tends to. Either 2 units over here and over here are highly active, and they silence these 2, or the symmetric opposite. These 2 are highly active, and they silence these 2. So what are these units that I'm waving my hands about? This one would be anti-ipsi on one side, anti-ipsi on the other side, pro-contra on one side, pro-contra on the other side. So the 2 stable states would be pro-contra on the left, anti-ipsi on the right, versus anti-ipsi on the left and pro-contra on the right. Okay, so that said, the idea is that when the light comes on and it activates one side of the superior colliculus, it would directly activate the pro-contra neurons on that side, and you would get the fast response. But if on anti-trials the anti-unit is a little bit more active initially, then the pattern that the network tends to settle in is this one, where the opposite pro-contra unit is the one that's active. But the time it takes to settle is much slower because it's no longer a synaptic time constant. Now it's a network time constant. And that explains the reaction time difference between the 2. Why is it that inactivating the superior colliculus would primarily affect anti as opposed to pro? And that's because we would also have an asymmetry between anti and pro. In the network, we built in an extra excitation for the pro units. That initially gives us, we see the asymmetries between pro and anti in 2 ways, both in the reaction time and then the percent correct. And so that means that it's more robust for the pro responses than for the anti responses. Once the choice period starts, it's settling into the attractor so strongly that inactivation during the choice period has no effect. That was one of the main things that we wanted to try to explain. Was that just a bunch of hand waving, or did it actually?
It was a lot of hands, but I get the general idea. It does require, the very specific patterns of inhibition between your 4 nodes and different settable weights on those. Not at all inconceivable.
Exactly. And we thought we could say, oh, this very specific pattern is a prediction from our model. Right. And it would be from that specific pattern of weights, but then we started wondering, but is that the only pattern that would solve?
So this might be very naive, but you're looking at the rats after 4 months of probably pretty intense training. Is this really learning? Is it just a bunch of reflexes the animal are using? And your output measure requires that the animal moves, so it must involve some kind of motor control too. So I'm not sure that you're basically, in a reflex, you're activating different networks. And it might be really interesting to look at the period where they're learning it, because during the learning you would require a different level of attention. If you have a task where they're all never failing, as in the pro, it must be a reflex. There's no thinking going behind that. You go into the mode and perform that.
I agree. That's what I was trying to express by saying, oh, here's the potential pro circuit, and it's so short, and it would be innate. That's why they would be able to learn it in a single session. The broader question of, would it be interesting to look during learning? Absolutely yes, we just haven't gotten there yet. And you also asked a question as to whether we think that it's learning or not. And I'm not sure I'm fully understanding you. I'm going to define learning as the animal's behavior changed after a certain pattern of stimuli and rewards. And by that definition, yes, learning definitely happens.
Sure. And how much do you think the motor, or the locomotion plays a role? Have you done this test in animal you paralyze? Would they activate the same?
That's a good question. We think that the superior colliculus is part of the motor control that is driving the animal to move one way or the other. That's, and that goes with those experiments where if you activate neurons on one side of the superior colliculus, you get contralateral motion. So we think that we're, near the top of the motor circuit. How this would affect how a paralyzed animal would behave in these circumstances, I don't know. The naive interpretation of what we're saying is that in the paralyzed animal, you should be able to record from the superior colliculus and see the same patterns of activity. Whether that's true or not, we haven't tested yet.